Okay. First of all, thank you, John, Becky, the CCP-EM team for inviting me and for organizing this fantastic symposium. It's very exciting to see this ever larger cryo-EM community in the UK and even beyond, and all these very exciting applications and methods, both in the posters and the talks. So today I will speak to you about new developments in Relion 3, and I thought it would be fun to show you, to remind you how Relion 1.0 GUI looked like. When I moved from Madrid to the LMB, I at some point decided I would write my own program, and I set out not to develop a package because I thought that was a lot of work, as I had learned in Madrid. So Relion 1 was just doing 2D classification and 3D classification, and that was basically it. So then over the years is 1.1 and 1.2, the kind of GUI stayed the same. There was a little bit of auto refinement added, etc. Then at some point, pressure came in, and it started to look again like a package that could do most of the pipeline. And then with Relion 2, I started a strategic collaboration with Erik Lindahl, who's also at the SciLive lab, as José Miguel just said, in Stockholm. They did GPU acceleration, which was a big advance in speed for Relion. And we also made this kind of the lower part of the GUI, which kind of keeps track of which jobs you do, etc. Okay. So 2.1, and now 3 was, again, the GUI hasn't changed much, but of course then the very big question was why orange? And this was decided in February 2018 when the Dutch women actually had bronze, silver and gold at the Olympics there. So then I thought that was a good excuse to do orange. Okay, so what's new in Relion 3? We'll go through that. Many of you have been using it for a little while, but I thought it would be good to show. And then near the end of my talk, I'll tell a little bit of what's going to happen in Relion 3.1. One thing is new, that until Relion 3, there was Xiaodahi, who was a PhD student in my lab, implemented helical refinement in Relion. But apart from that, all the Relion developments were just done by myself. So one important thing that's new for development of Relion 3 was that these 3 people joined my group. So Jasenko Zivanov came highly recommended from Henning Stahlberg from Basel, and he's now left us again. He's gone back to Basel, which he thought was very nice for living. And he has developed Bayesian polishing, CTF refinement, Beamtilt estimation, and some other stuff that I will be showing for Relion 3.1 as well. Then Takanori joined us from Japan. Garib and Andrew Leslie had worked with him, and he came out highly recommended from them. He's been involved in multi-body developments and testing, many tweaks already, and I'll show some of his developments as well. And as many of you will know, he's now replaced most of me on CCP-EM in replying to lots of questions, and he's incredibly patient, much more than I am. So, and Li Yidong is now more or less halfway into her PhD, and she's working on automation of 2D class selection using a combination of machine learning and feature-based methods and so on. I will not show her your work yet. It won't be part of 3.1, but I'm very optimistic it will become part of a well-functioning part of 3.2. Good. Then, besides the GPU acceleration that Erik did already for 2.0, in 3.0, Erik and his team also expanded this work, kind of made use of the same unwrapping of all the loops and put them all in large arrays for very parallel calculations to do acceleration by vectorization on CPU. So especially if you have these relatively modern Intel compilers and the Intel chips, then this will lead to significant speed ups. In terms of price to computation, they can't beat the gaming GPUs yet, but now you can run a CPU more or less as fast as you can on these double precision GPUs for more or less the same amount of money. Okay, few new features on the Relion 3 GUI. One is the own implementation of the Motion Core 2 algorithm that was developed in David Agard's group. And this is done by Takanori, who is even more outspokenly open source advocate than I am. So he was not happy with Motion Core 2 being closed source. And also, because of the Bayesian polishing that Jasenko was developing right next to Takanori, we needed more integrated access to some of the metadata of the actual whole frame movie alignment. So Reliant Core 2, as people now call it, is open source, free for all, no license issues or so on. It's currently only CPU, and that's because we see now multi-threaded version. If you use a relatively modern CPU, we don't see much gains over GPU. Perhaps this balance will change a bit again with K3s, which are bigger, and then we might reconsider this. But we get more or less similar speeds as the UCSF version for the 4K by 4K images, and then we get improved passing of the metadata to the Bayesian polishing. That does not mean if you use the UCSF GPU accelerated version of Motion Core 2 that you cannot run Bayesian polishing. That's still fine. Okay, now on the auto-picking, there was a few new options. One is just a bit of convenience, really. You can now provide a 3D model, and then it will make projections in all different orientations and use those for template-based matching. And we also added a Laplacian of Gaussian auto-picker approach to be able to start your auto-picking without any references. This just looks for blobs of a given size. It's very similar and inspired by Dog Picker, which was developed, I think, in the Carragher and Potter labs. And I don't have any slides. I'm going to mention that this is still an active component of our research, and we're very much inspired by the work in Sphere by Torsten presented just now, or Dmitry Teganov and so on, and we'll see how in the future we can continue to improve auto-picking in Reliant. Okay, then 2D and 3D classification have this nifty little new option which is called use fast subsets. And this is an idea that we nicked from Nico. And I think this is the beauty of open software. And Nico is also a very outspoken open software advocate. And we all kind of have different ideas. And we all kind of keep using each other's good ideas such that all together we can progress science a lot faster. So this idea comes from System, where if you do 2D or 3D classification with 1 million particles, in the first iteration it will be either circular little blobs for the 2D classification or very blobby 3D reference in 3D, and you pass along 1 million particles, which takes a long time before you then update it to slightly less circular or blobby 3D. So the idea is we use just a few particles in the early iterations, make sure the circles are no longer circles, but start to acquire some features with not so many particles. And there's just some heuristics in that, in how quickly you make that happen, which seems to work fine. And if you do have 1 million particles, this now runs up to 10 times or even more, even faster than that, than the original approach. If you only have like 10 or 15,000 particles, then this wouldn't matter much because it's in the range of a few thousand particles that you're using in the first iteration anyway. Okay, then on the initial model generation tab, there is now a lot more options, and they mimic the options of the CryoSpark program more closely, and that's where this program originally comes from. That's where open source stops being in place. But so this is work done by Marcus Brubaker and Ali Punjani, as John Rubinson already mentioned yesterday. So this now works a lot better in Reliant 3.0 than it did in Reliant 2.1. Then the first main algorithmic improvement, which is a lot more work than the ones I've shown until now, is multi-body refinement. And this was started by myself, mostly kind of prototyped by myself, and then when Takanori joined, we took on this project together. So multi-body refinement is an automated way of doing focus refinement if you have multiple independently moving or flexible domains in your large complex. So just to refresh you, focused refinement with partial signal subtraction. We first presented this in an eLife paper in 2015 on gamma-secretase. It was kind of independently developed by Yuha in Oxford and also by Hongwei in Tsinghua University in Beijing. But basically, if there's one part of the complex you're really interested in, what people would have done in the past is to put a mask around that part of the complex and then do your 3D refinements or your 3D classifications based on projections of that masked reference. Now, if you have masked reference, for example, you would be interested in the red part here, this gamma-secretase, you could mask that out. Then your reference projections only have density from the red part, whereas your original experimental particles have the entire gamma-secretase density in them. So if you now compare this one with this one, then the yellow part of gamma-secretase just adds as additional noise to the comparison. So what you can do, you can actually subtract that part of the density and have a more consistent comparison. So that's what focused refinement is. And one early application was done by Kelly Nguyen in Kiyoshi Nagai's lab, now at Berkeley with Eva Nogales and Kathy Collins. And the spliceosome is kind of one stable core, and then they call it a head and an arm and a foot, and they all independently wiggle with respect to the stable core. So you can, what she then did is subtract the stable core, the arm and the foot, and then focus refinements on the head and made that better, focus refinements on the foot, made that better, etc. That was a lot of work and a lot of expertise from Kelly going in there. So the idea of multi-body refinement is to do this in a more automated, iterative manner. And I will use first a kind of a dummy problem to explain this. So this is a 3-domain floppy protein, which is called MRC. So there's the M domain, the R domain, and the C domain. And if we imagine this just kind of 3 beats on a string, which kind of, there's floppiness between the M and the R and the C. The way it works is we define, as a user, 3 masks of these 3 domains, and we have MRC images, and we have some kind of consensus refinement where we've put them all together. So the M will be blurred away and the C will be blurred away, whereas perhaps the R is kind of okay. We can make references with only the M because of these masks, and then we can subtract the R and the C density from this image to have an image which hopefully only remains the M. But because the consensus model isn't perfect, that subtraction isn't perfect, so you can say we've done kind of okay job, but there's still some remaining density. What you can now do is this alignment of the M, this image being aligned to the reference projection only containing the M, and you can now keep the optimal orientation of the M domain for that particle for the next iteration. You do also the R domain and the C domain in the same way, and then the next iteration, the partial subtraction of each of these domains, because the relative angles are now specific for this individual particle, will be better. And you can keep doing this until things converge, just like in normal refinement. So, as an example, I'll show you, again, a spliceosome. This is a B complex, which was done by Clemens Plaschka in the Nagai group. And Clemens divided this spliceosome complex into 4 a kind of major independently moving part. The core, the foot, this is called the helicase domain, and the SF3B domain. And you can already see that the density in the SF domain is rather poor, the helicase is not great, and the foot is also a little bit worse than the core. Now, in a consensus refinement, where we put the entire data set together, because these domains are kind of flopping around, you can see these, this is the core, and then these are different heights of the slices through the map. You can see here the foot slightly worse than the core. The helicase domain here is a lot worse than the core. And the SF3B is basically one just kind of grayish blur. There's no way you're going to build anything into that. Now, by iteratively refining these relative orientations of the 4 domains while subtracting the other 3, we now get 4 new maps. They're independently written out, independent MRC files of the core, the foot, the helicase, and the SF3B domain. And now you can see kind of molecular-like details in all of the maps. You can You could quantify that with local resolution maps, and you can go from maps which are really not interpretable at all to maps which are, at least here, interpretable, and then here kind of getting there. And FSC curves improve dramatically. Now, what you end up with is for the 100,000 particles in this data set, each domain now has 3 relative orientations. Each domain has 3 angles which describe its relative orientation to the consensus model. Now you can do a principal component analysis on these relative orientations for all the different domains of all the particles, and that you can get an idea of what is the principal motion in your data set. And you can make histograms of what the explained variance for each of these components, and then you can make movies where we now position these rigid body maps along these motions that we've identified, which are the most important one in the data set. And this is the second motion that Kiyoshi, for some idea, thought was, for some reason, thought more interesting than the other. For example, you can see that the SF3B domain very often kind of rotates together with the helicase domain. And these rotations are much bigger than the ones that occur in the foot, explaining why these were all kind of more or less blurred out in the beginning to start with. So not only do you get improved density for flexible paths, and you can do some atomic modeling, but also you get an idea of what kind of motions are present in your sample. Good. Now we get to work done by Jasenko. And the first thing Jasenko did was to have a look again at beam-induced motion and how we can correct for that. And he came up with this Bayesian polishing algorithm. And it completely replaces the particle polishing that was present in Relaun 2, because this works better in all the cases that we've tested. So we just like in our normal refinements, we express the problem as a regularized likelihood optimization problem. We're going to try and find the most likely tracks for individual particles inside these direct electron detector movies, given some regularization terms. And these terms are similar, as John already mentioned yesterday, to things that he also uses. So you can't be accelerating too quickly, and you can't have a too rugged track. They should be relatively smooth. But for example, the old polishing approach, the speeds were supposed to be linear, and you would only do linear velocity as well. Now we can do actual curved tracks, and there can be some acceleration, and the user can control how much there is. Now, just to show you some tracks that then come out of this program for plasmodium ribosome, for beta-gal, for gamma-secretase, a lot of stage drift in this one, but also what we often see the initial motion, which is where the little blue dots can be a lot stronger in a different direction than what the final motion of the whole frame settles into. Now, this is all beautiful, but what does that mean in terms of results? So, at some point in time, there was motion core 1, and then if you would do particle polishing after motion core 1, you would get huge improvements in resolution. Then came motion core 2, which already did 5x5 patching, and part of the advantage of polishing was lost, and there was not so big a difference between particle polishing and motion core 2. Now, again, there is an advantage in going back to the individual particles in Bayesian polishing compared to motion core 2. So again, we go, for all the test cases, we increase from our own motion core 2 or UCSF motion core 2 to the Bayesian polishing in resolution. And perhaps more important are that we get improvements in B factors. And kind of to cut a longer story short, if you do Bayesian polishing, you kind of need 20 to 30% less particles to get to the same resolution. So if it's all about throughput and time and money, then that's a good. If FEI would give us this discount, it would be good, I think. Good. Second main contribution of Jasenko to the Relaun 3 release was this new job type called CTF refinement. And CTF refinement contains of 2 different parts. One is, again, not a new idea at all. I think Nico already did this in Freeline many years ago, but to refine the defocus of individual particles by comparing it to the reference projection of that particle. Now, it's very important, we've seen, that you maintain your gold standard separation of the two halves, because there's so many parameters to introduce here that if you don't, you will definitely overfit your data again. And then the second aspect of this is Beamtilt estimation. And I'll come back to that. About why it is a good idea to do defocus estimation for each particle by comparing it to the reference, rather than only relying on defocus estimation from the micrograph. The micrograph has an advantage, it's a lot bigger area, so that contains more signal. But it's only the power of the tone rings, and whereas if you look at the reference projection, you know the power of the signal, and you can look at square differences. And the power of the signal compared to how much noise there is a lot better if you do these per particle comparisons. Although, of course, the particle itself is a lot smaller. So we tested this on a kind of extreme data set just to show that it works. And this was Empire 1097, I think, by Dimitri Lyumkas, if I remember correctly. They did 40-degree tilted image acquisition to get around the preferred orientation. And that, of course, leads to a very steep gradient of defocus within each micrograph. And because it's so steep, it kind of goes with outside the rates of convergence of one single CTF refinement. But we've seen if you now do this multiple times in a row and you combine it also with some Bayesian polishing in between, then we can get to a resolution of 3.1 angstrom, which is considerably better than the 4 angstrom or so which it was published originally. So per micrograph defocus is kind of a very tricky thing with these highly tilted images, but in the end you can get to a nice high resolution model. And if you then look at individual micrographs, how the defocus changes, you can see that most of the particles have kind of for most of the particles has found the right defocus. This is not to say that this is the optimal way of processing your highly tilted data. I think it would be better to use something like CTF tilt or something that actually can deal with tilted micrographs at the micrograph level and then perhaps refine them afterwards. Then you don't have to do all these different steps. But it just shows you that the program can do this. So the second part of this CTF refinement job type is Beamtilt estimation. And now we come to relate to Sonja's talk, so I can kind of skip this. You normally focus and take one image or take 4 images, and now the idea is to take 9 holes, for example, without moving the stage, and it was all explained by Sonja. And this is all in very close collaboration with Wim Hagen at the MBL in Heidelberg, because he collected this Apo-ferritin test case when he was testing his cryo-EM scripts, where he had been playing with this active beamtilt compensation, which you called aberration-free image shift. Yes, thank you. So he did 5 images per hole and then 9 holes before you move the stage and repeat that 9 times. So you had 405 images with this active beam tilt compensation on inside the microscope. Now, that's probably, if that works well, that's probably your preferred way of doing your experiment, because I always think you should do your experiment as well as you can. Now, if you can't or don't want to fork out the upgrade, you could do this without, but then you would introduce a lot of beam tilt because of the beam shift introducing the coma. So the good thing, of course, is you go a lot faster, and he did this experiment already 2 years ago, so now at 130, or a year and a half ago, at 130 movies per hour, which was a huge improvement. So if you, and this is now in response to a question that you got, if you do the active beam tilt compensation, then this is your FSC curve, which was, I think, that's about 2.0, 2.1 or so angstroms resolution. If you did not get the upgrade, or you decided not to switch the option on in Wim's case, then this is your FSC curve. So your map actually, and it's still 2.7 if I remember correctly, so it's still a nice map, but the difference between the green curve and the gray curve is the effect of the beam shift induced beam tilt. And if you now estimate for each of these 9 holes, we divide the data up into 9 subsets, which is hole 1 to 9, and the middle hole will have almost no beam tilt, but the center, the diagonally away holes will have a lot of beam tilt, and we estimate beam tilt from comparisons of the reference projection with each experimental image. We see what is the phase difference between the projected reference and the actual experimental particle, and that we average over all the particles in that subset, and we model that with the beam tilt. Then we can get to the purple map, which is just as good, indistinguishable really from the active beam tilt compensation. So the answer to the question you got is yes, you can do this in the software. You can estimate how much beam tilt there is and correct for it, and that is as good as doing it in the machine. Now, as I said, I think you should always do your experiment as well as you can, but it turns out we've been running this on many data sets since Reliant 3 is available, and everybody does coma-free alignment in this room, I hope, if they use a cryos, but there is a surprising amount of data sets which have a significant amount of beam tilt, and I don't know where it comes from, if it's user expertise. At the LMB, I think our Cryos 2 tends to have higher values than Cryos 1 and 3, so there might be something with kind of preset alignments. I don't know, but these are observations that we make. So you shouldn't need this, but if you do have some beam tilt, it's actually quite nice to then suddenly get another extra boost of half an angstrom in resolution. So these are plots of the average beam, the average phase difference between all the experimental particles and the reference projections for these 9 different holes. You see in the central hole it's more or less 0, but if you go away, if you shift the beam all the way over here, then in the direction perpendicular to that beam shift, you get all these beam tilts, and this goes to the resolution to the cube, so that goes pretty hard. And depending on how much you have, this can really damage your resolution. Rafael from Madrid told me he'd collected the data on a cryos, and it got kind of stuck at 4. He said, this is a really good sample, should go a lot better. And it was not Apo-ferritin, it was a real sample. And then he estimated beam tilt corrected for it, and suddenly had a 3 angstrom map. So there was a lot of beam tilt in that data set, and just by correcting for it, he completely salvaged the data set. Good. One of the things, and other people have already told about it, is this on-the-fly processing. So we've made a command line, and thereby Python-accessible interface to how you submit jobs on the GUI. That previously was only possible by hitting the run button. Now you can actually prepare a job offline and do a command line argument that then submits a job to the same pipeline. So you can write Python scripts, that's what we did. Takanori and I kind of played around with this, and we hope it serves as inspiration. And already Sean Rawson, who's now at Harvard, kind of demonstrated that's true, that people will now change these and modify them to their own needs. It just automatically runs motion correction, CTF refinement, etc. And then you can just open your GUI and everything is as normal, only that you didn't have to click on all those jobs. So to test this, we thought, let's not use gamma-secretase, because everyone can do can do Apo-ferritin. So we thought, let's do gamma-secretase, which at the time was quite a hard structure to crack. It's still one of the smallest membrane complexes out there by cryo-EM. This was done by Sjors Bai, who is a very good, very experienced postdoc in my lab. He's now running his own lab in Texas. And what we did is, this is the map, the gray line is the FSC curve that Sjors got out in Reliant 1.3 back then. And if you now use Sjors very carefully selected particles from multiple 2D classifications of subsets of the data and reclassified, and everything very carefully curated, and then just run Reliant 3 with Bayesian polishing and CTF refinement, etc., then you get the green line. So you get an improvement from 3.4 to 3.1, which is just because you upgraded your Reliant installation. So what we then did was to run relyonit.py script, and that script automatically pick the particles, and then what it will do, it will do an initial 3D model generation automatically, select what's hopefully the right one, and then classify a 3D classification, and then make projections of a preliminary 3D model for template-based picking, and use that as your particle set. And if we now use that particle set, and now it stopped being completely automated, we now just manually went into refinement and basing, polishing, etc. But if we used the automatically acquired data set, then we got to about 3.2 angstroms resolution, or so 3.2 or 3.3. So it's not as good as Xiaochen yet, but it's getting there. And then our kind of show-off little structure was already mentioned before, I think yesterday. This was Apo-ferritin for Wim, which was the champion structure in terms of resolution on Twitter for about 2 hours. Very proud of that. Which then went to 1.65 angstroms resolution, which is actually very, very close to the Nyquist frequency, but I think the features in the map validate the resolution claim. Good. Last part of the talk. What is new in, because hopefully you all knew about this already, what's new in Relion 3.1? And well, the GUI will be pink. That's because my wife's called Rosa. There won't be that many new functionalities as there was from Relion 2.1 to 3.0, and I'll walk through the most important one. This is a minor one, but it's kind of an important source of annoyance for many Relion people. Some of the more adventurous would use UHA to do better subtractions of things that were not centered in the center of the original box, or to get subparticles into smaller boxes, etc. So that has been dealt with in Relion 3.1, so you can do your focus classification by recentering particles in any point 3D space of the original consensus refinement, and you can rebox it, etc. So that's just to solve some annoyances. The main source of changes is in the CTF refinement job type. And now we have again the particle defocus and so on, and the Beamtilt like we had, but we now have a new set of options, which are these higher order aberration corrections. Now, what are those? There are two types of higher order aberration or kind of aberrations in general. There's the asymmetrical ones, and the phase shifts that you get from the Beamtilt are in this category, and threefold astigmatism, which I will show you in a minute, is in this category as well. Then there's the so-called symmetrical aberrations, and these are actually, they're not shifts in the phases of the CTF, but they're shifts in the argument of the sign function of the CTF. So we change these as well, and these actually make your CTF rings no longer round, but slightly squarishly shaped. So this, and also CS optimization, you could consider symmetric. CS is also a form of symmetric aberration. Fourfold astigmatism is another example. Now, because this has kind of gone right into Relion's heart by changing CTF model and so on. So the kind of coding involved and how your metadata will be arranged in star files and so on will have significant changes between 3.1 and 3.0, and that's why it's all taking much longer than I had hoped. But what we do is we model. We again measure these differences in phases with respect differences with the reference projections, and also in the argument of the sign function with respect to the reference projections. And we model these using Zernike polynomials. So whereas the kind of Beamtilt model had a nice physical meaning, now there is no longer physical meaning. There's a linear combination of Zernike polynomials that tries to model what goes on with your data, but there's no real direct interpretation in terms of physics or optics. But it does work. So our most impressive example thus far is from Empire. This is data by Gabe Lander and Mark Hertig and Menu Wu at Scripps in San Diego, and they collected this 20S proteasome dataset already quite a while back on their Talos Arctica. I think this was in their early days of Talos Arctica, and Gabe has, of course, gone on to do all kinds of amazing things with his Talos. Now, in Relion 3.0, we got this structure to about 3.1 angstrom. And when we now looked at the phase difference, the per pixel phase difference plots of the Beamtilt estimation, then we got this picture. That's another new feature of Relion 3.1. You can do color images. So we get positive and negative changes in phase shifts, no longer on one side positive and the other side negative, which would be Beamtilt, but this has this kind of threefold, pseudo threefold. It's not entirely threefold. There's a bit of a twofold component, which is the Beamtilt, combined with a very high threefold component, which is this threefold astigmatism. So this is the model that we use from the just a linear combination of the Zernike polynomials to describe what's going on here. And now, when we refine our particles, we just apply these phase shifts to the data, no longer really caring what the physics behind it is, but just knowing that this is what happens in the data. If you look at the arguments of this sine function, how they differ, there's this distinct fourfold symmetric aberration going on, or people have called this tetrafold or fourfold astigmatism. And this is the kind of model that Jasenko estimates from these measurements. And by doing that, you can improve this 3.1 angstrom structure into a 2.2 angstrom structure. So that's nice. Now, then we asked Gabe, what did you do with your Talos? And this was already quite a while later than that we got these results, then he collected this data set, and he said, I don't know. And then we said, okay, can you have a look at, because by then he'd done all kind of high resolution structures on this Talos, much higher than these 3 angstroms. We said, can you have a look at these images that Relion 3.1 already wrote out and see if there's anything like this happening? And he said, no, it's gone. I said, oh, did you do some kind of special alignment or so? And he says, no, I don't know. So still we don't really know where it came from. It seems to be gone now, but yeah, it's slightly disconcerting that we don't really know what happened there. But at least the good thing is, if it does happen, you can now correct for these things. So then you say, oh, okay, 26 says 20S proteasome is just another Apo-ferritin, is just another beta-gal, is ultimately just another ribosome. That's where we all started. So that's just to show you that this is also useful in real life. And this now goes to my other hobby, which I've recently acquired, is to look at these tau filaments, which this protein tau, which normally stabilizes microtubules, then aggregates into these amyloid-like filaments in a range of different diseases, Alzheimer's disease being the most common one. But if you're engaged in professional boxing, or perhaps you like American football and you like to bang your head into others, and you're unlucky enough to have repetitive brain head trauma, then you might be very unlucky and develop this disease called CTE, chronic traumatic encephalopathy, which is it's a devastating neurodegenerative disease where you get character changes and dementia and then you die. So we were looking at these, and we had a very successful trip to Diamond, collected data on their Cryos 4, and then when we had a look at this plot again, we again could see threefold. And the contrast of this one isn't so good, but it goes all the way to 1 rad beam phase shift at the edge of the resolution here. And by correcting for both 3 and 4-fold astigmatism, we could improve this map from 2.7 angstroms to 2.3 angstroms, which then allowed us to see water molecules, which no one had ever seen inside cryo maps of amyloid. So there's ordered water molecules. We could also see alternative side chains, which kind of explained how alternative positive and negative charges on the outside of these filaments can kind of do it with each other in alternating ways to compensate for all their charges. So you can learn new biology with this as well. And we're still finding data sets where this is a problem. This is not a problem at 4 angstroms. It might not even be a big problem at 3 angstroms. But as soon as we go and push this thing towards resolutions that Pamela here from Astax was happy with yesterday, then all these things might become important, and I think it's good to monitor these. One last thing is magnification refinement. How are we doing? We have to catch a train. Relyam 3.1 will be able to refine relative pixel sizes because you know your nominal magnification, but perhaps not that well. You'll also be able to do this in the different X and Y directions, so you can do anisotropic magnification, etc. This will also allow you to combine data with different pixel sizes and box sizes all into one refinement, and then hopefully have it all sorted out for you. I don't have data yet. That's the part that's holding us all back. That will mean you'll need to import your data into different groups from different detectors, etc., which will be a new import. Oh, that one is there. Okay. This will take some time. It's not even properly alpha yet at the LMB, so we're hoping to go beta somewhere in the fall, I think. Please use CCP-EM email list to send us questions. I still get several a week direct questions, which I tend to delete, so I'm sorry, but I don't have time for that. And then, on another note, if you'd like a permanent position in a very exciting team and love electron microscopes and would like to give support to our very exciting EM community at the LMB, then we'll probably soon have a position open. Just contact one of us. Thank you very much.
Questions?
I have a couple of questions related to the rely on it and 2D classification. So you did a test given to someone else, not in your group, how it performed compared to, I mean, not in your hands, because I guess you set up some of the parameters for picking, and the other ones, maybe I'd say both, and you... for 2D classification, at least for cleaning and the automation, I have seen like the classes doesn't change that much between iteration 15 and 20. Have you have any numbers or evaluation about that, at least for cleaning, not for the final 2D classification?
So multiple people have been running relayonnet.py and seem to be happy with it. I'm aware that picking needs to be improved. So 2D class selection automated is not yet part of it. That's why the current script uses 3D classification. And it just happens with a default number of 20 iterations. And if that's more than you'd needed, then you've just wasted some computing time. But we kind of try and put the parameters such that we can deal with as many cases as we want. But as you also know, it's quite hard to cater for every need. But yeah.
Maybe I ask one. Your multi-body refinement at the moment requires that the user identifies the 4 bodies in the example you showed. What are the possibilities for defining the bodies and how it's separated out?
So Takanori's real project is exactly that. Also, each body needs to be at least 100, 150 kilodaltons for it to be able to align. We're working on making much smaller bodies, but which speak to each other such that you can do this in a more continuous way, and you get rid of this very user-intensive step of defining these different masks. But that's not ready yet by far. Hugo there, shout.
Thank you. I really like it. Looking forward for the next Reliant 2.1. So, if I remember well, for polishing, you track movement by micrograph. Sometimes there are not many particles in micrograph. How many particles do you use for polishing?
Right, so the question is how many particles should be on each micrograph to do polishing. That all depends on how large your particle is. If you have ribosomes, they can be tracked very well individually, I think. If they're gamma-secretase, you'll need many more on a micrograph to have reliable track, so it all depends on the size of the particle.
Hi, what do you think about the idea of solving for rotations between the frames? Do you think that's a particularly useful avenue to go down, or do you think it wouldn't bring that?
Well, I did in the past. So before polishing, there was movie refinement, which did exactly that. So each movie frame was also rotationally aligned. What we found was that the accuracy with which you could refine even subsets of frames quite large was much... the actual rotations that we observed for different particles were much smaller. So for most particles, you will not be able to do that very accurately. Perhaps if you have very high resolution and very nice big particles like viruses, it might still be worth it. But at some point, we chose not to do that. It makes the entire pipeline very, very expensive. And for most cases, it's probably not worth it.
I think we're going to stop there because I know that people have trains to catch, including Sjors and I. But there are a few administrative things to do at the end. But first, let's thank Sjors again. Okay, so Becky and I had a lot of fun putting together the program and selecting the speakers and going through the abstracts and things. We hope that you enjoyed yourselves. And there are a number of people that need to be thanked. The first are all of the speakers for coming and talking to us. So thanks again to all the speakers. The second are the people that actually organised the meeting, because really all we did was select the speakers. And you've already seen quite a bit of Tom and Colin, who've done a lot of work behind the scenes, so I think we should thank them. And then a huge chunk of work from Helen, Karen and Bryony. So thank you very much for making the meeting work. And we have flowers. So, next year, the meeting is on the 28th and 29th of April, so the same time, and it's in the same place. It's here, as you've already heard. This has been a very nice venue. Some of you may have noticed that I did the cooking, and I won't do it next year. Sjors and Tom have volunteered. There will be a chance to give feedback, and I think the CCP-EM team will be very grateful to get that feedback. And I understand that Helen, I think, will send an email to all of you to ask for your comments on the meeting. So send that. And then finally, thank you all for coming, and hope to see you next year.